All right, everybody. Welcome to the first 2025 MycoTalks. My name's Jay Coles. I'm happy to co-chair this seminar today with Dr. Ilyev, and we have two outstanding speakers, Dr. Teresa O'Meara from University of Michigan, who's going to talk on understanding Candida albicans during healthy human colonization, and Dr. Chad Rappleye from the Ohio State University, who's going to talk about the metabolism of the fungal pathogen Histoplasma capsulatum, enabling intraphagosomal proliferation. So just to give a background, I welcome on MycoTalks. So this was actually started during the in 2020 during the COVID pandemic, and the philosophy and origin is to foster diversity, including career stage, gender, geography, and field of mycology. We have two 30-minute talks on all aspects of medical mycology, followed by 30 minutes of in-depth discussion. Please submit the questions through the Q&A tab on Zoom, and Dr. Ilya and I will field those questions and bring them to the respective speaker. And in these sessions are recorded, and they'll be available on the MRC CMM YouTube channel. Next month, we have Dr. Elisa Gerstein from University of Manitoba and Dr. Stu Lebitz from University of Massachusetts Chan Medical School. And then in March, we have Dr. Rebecca Shapiro from University of Guelph in Canada and Guillaume Jambon from Institut Pasteur in France. In addition to the MycoTalks, we have the Medical Mycology Training Seminar Series. This is for students, postdocs, and clinical trainees, as well as new faculty to present your work virtually to a global community and get feedback. And these talks are on the second Thursday of the month, but timing can be adjusted if time zones are an issue. And this series helps us highlight a scientifically broad and international medical mycology in the community that's generating new science. And sorry. And below is the link that to submit your abstract if you're interested in participating in this series. And then also we have MycoClinics. The next one is scheduled for March 6, 2025 at 4 p.m. UK time, and it's on the management of rare mold infections. And this is a quarterly series, and the organizing committee is listed down below. So with that note, I'd like to introduce our first speaker, Dr. Teresa O'Meara, Assistant Professor of Microbiology and Immunology at University of Michigan. Dr. O'Meara went to University of Chicago for her undergraduate degree and then followed by a PhD with Andrew Alspaugh at Duke University, and then a postdoctoral fellowship with Leah Cowan at the University of Toronto. And she started her lab, I believe, in 2019 at University of Michigan, and studying both Candida albicans as well as Candida auris and understanding virulence and fungal pathogenesis. So it's my pleasure to introduce Dr. O'Meara, and we greatly look forward to her talk.
Everybody, just get my screen up. I am super excited to tell you guys about some of the work that we've been doing in my lab to understand Candida albicans during healthy human colonization. And so first I want to highlight this work was really driven by a fantastic graduate student in my lab, Faith Anderson Davis, with help from a technician, Noelle Visser, who is now getting her own PhD at the University of Louisville. And the main topic we want to understand here is that Candida albicans is both a leading fungal pathogen causing hundreds of thousands of serious invasive systemic infections per year, but it's also part of the natural human mucosal microbiota. And so we have this dichotomy between strains that can cause this invasive disease and then strains that are within us all the time. And so we wanted to ask the question, are the strains that cause disease different from those that colonize? And can we use this information clinically? The reason why we wanted to take on this topic was because a model had been proposed that there are fitness trade-offs between disease and commensalism. And this was driven by some really nice studies in mouse models where they took SC5 and 4, which I'm going to refer to a lot as our clinical reference isolate, and experimentally evolved it in the mouse GI tract. And at the end of that experimental evolution, we end up with non-pathogenic isolates. Or in a functional genomic screen, we're identifying genes that are required for colonization. We again saw that trade-off between filamentation and colonization in the gut. So strains that we're colonizing tend to be afilamentous and avirulent. So we wanted to know, do we see that same system when we collect strains from, not from mice, which are not a normal host to Candida albicans, but from humans? And so this is actually the first experiment that I did in my lab. So we took sample collection from healthy undergraduates. There's actually a cool course at Michigan called Bio 183 where they sample their own microbiota. So here the students were already donating fecal samples, so it's not too much of a trouble to ask them to donate a little extra for us. And then we're able to give them chromagar plates so they could swab their own mouths, and we could pull out different colonies from them. So here's a picture of my healthy undergrad, Katie, collecting all of those isolates from our healthy undergrad samples. And I also want to emphasize that we didn't pick just one isolate from each student. We picked every single colony on that plate. And so we had some students that had just oral colonization, some that had just fecal, and some that had both oral and fecal colonization with Candida albicans. In general, we got more colonies from the oral samples than the fecal samples, but there's only so much you can get from a gram of poop, so we'll go with it. And throughout this talk, I'm going to have all of the samples that were from the fecal isolates being in pink. So if you want to remember, it's pink is poop. So given that said, we had almost 900 isolates from these students, and we saw a huge amount of variation in their ability to grow under different conditions. So here what I'm showing you is the growth rate, so how quickly they're dividing in growth in either dextrose, galactose, or glycerol, so pretty standard carbon sources. And we can see that there's a lot of variation between these strains. And we also see that they vary in their carrying capacity. So this is the maximum OD that they can reach in these different glucose conditions or different carbon sources. And again, you can really see this exacerbated for YPD or dextrose, where there are some strains that grow really well and can reach a high carrying capacity, and some strains that are not growing very well. And again, these are all isolates that were collected basically from the wild, from a normal human microbiota. These students were healthy enough to go to class, so I'm going to call them healthy undergraduates. And this says that there's not like a standard feature where they can all grow very well in all the different conditions. We also screen them for their ability to invade agar. Agar invasion is often used as a proxy for filamentation. So we looked at their invasion into regular YPD agar under different stress conditions and spider medium. And what you could see here is that there's a huge amount of variation where we have some colonies that have this huge halo of invasion and other colonies that are really limited in their ability to invade that agar. But invasion is not the only measure of morphology. We can actually look at the cell shape through microscopy. And so what I'm showing you here is in the non-inducing Q of YPD at 30 degrees, we can see that the reference strain S5E4 grows as these little yeast. And then when you move them to a inducing Q, such as 37 degrees with 10% serum, now we have these nice long hyphae. But when we looked at all of our commensal isolates, we saw an interesting pattern in that they all behaved like that reference, and they all could do that transition from a yeast form in the non-inducing Q to a hyphal form in the inducing Q. Sometimes the hyphae looked a little different, but they all had that ability to do that filamentation growth. So this suggests to us that unlike in that mouse model, the human gut isn't consistently selecting for yeast-locked strains. And I also want to point out that we didn't identify any isolates that were constitutively filamentous in the non-inducing Q, which again is different from a previous study from the Cowan lab that has shown that in cystic fibrosis patients, there's a repeated selection for constitutive filamentation. So I think this really emphasizes that in these different mouse models or human models, we have different selective pressures on morphogenesis, and there's different programs that are going to be involved in each of these different environments. We then took all of our phenotypic data that we'd done on all 900 isolates, and what we saw is that there's not a clear separation in strains from different isolation sources. So here, this is a UMAP sort of clustering all those phenotypes together. And in pink are all the fecal samples, and in black are the oral samples. And there's not really a separation, right? The pink are sort of well embedded within all of the clusters here. So there's not a separation, which is a little bit surprising to us because we would assume that the oral environment is different from the GI tract, especially all the way down to fecal samples, but it's possible that there's a continual recolonization from the oral tract throughout the GI tract, and that's why we see some of those same phenotypes coming out. But we also tried to separate these out and color them not by the sample source, but by the sample by the student source. And so here they're all colored by what student the sample came from. And again, we don't really see a clear separation. Like there's maybe a little bit of clustering of related strains from one student, but it's not like a clear clustering away from each other. So potentially the range of phenotypes that are observable in people are sort of well scattered amongst all of the different strains. Okay, so here I've told you about all 900 isolates. We want to dive into like, get a more detailed analysis. So we selected a set of 46 isolates from these different students based on whether we had matched fecal and oral isolates from the same host. So can we see, this will allow us to identify that diversity within an individual in the different environments. And then strains that showed variation in growth or invasion in the initial screen. So we're trying to select for the most differences that we could identify within a strain. So using that, we started by doing whole genome sequencing, and we placed them in the context of all of the other Candida albicans strains that had been previously sequenced. And this is work that we collaborated with Tim James and his student at the time, Kevin Ames. And so here what I'm showing you is a phylogenetic tree of all of the different Candida albicans isolates, and they're colored by these like defined clades that are from the Ropars data set from Christophe Donfer's lab. And we sort of place our isolates in the context of these previously sequenced strains. So we do have representation from multiple different strains and clades. And then just as a reference, here's SC5, 3, and 4, our reference strain. What we see is that the U of M students have isolates from across the phylogenetic tree. So we're not just getting clade 1 isolates, we have isolates from across the tree. We also see that some of these students are colonized by multiple clades of Candida albicans. So potentially there are multiple introductions to that student. So we have one student that has representative isolates from clade 1, and then the closest relative clade is clade A or C here. And so potentially people are colonized by multiple strains, which gives rise to the potential for intra-strain competition within the host. We also see that some people can share their strains. So here we have student 811 and student 882. And these are these 4 isolates. And based on the branch length, we basically can say that these strains are highly related to each other. And it's very likely that these students are sharing their Candida albicans. Maybe they're in the same lab group, maybe they're roommates. But it's clear that they can be transmitting strains between each other as well. Not only did we look at the sequence variation, but we also looked at the structural variation between the strains. And so here what I'm showing you is a chromosome chef gel. We're separating out all the different chromosomes. We have our SC5, 3, and 4 reference here. And we've marked where we think the chromosomes should be based on that SC5, 3, and 4 reference. And I've indicated in some of these arrows where the sizes don't really match that SC5, 3, and 4 reference. And so we did this for all of our strains. And we end up with a huge amount of variability. So here I've ordered the strains by their genetic relatedness due to their SNPs and their phylogenetic relationships. But what we can see is that there are certain strains that have an extra sized chromosome. Potentially there was a loss or a recombination event. And this is consistent across all of them so that we can see extensive structural variability between the strains as well. Because we had sequenced the strains using a modified synthetic long-read sequencing, we're able to identify some of these genomic rearrangements. So we don't see a huge amount of aneuploidy in these strains, but we do see that not all of the chromosomes that we know from SC5, 3, and 4 are matched when we make our contigs using the TEL-seq synthetic long-read sequencing. And I want to show you here, these are the different chromosomes, and with the TEL-seq pipeline, with our SC5, 3, and 4, just to say that, hey, we can reassemble most of the contigs that we were expecting to see. But in some of these different isolates, even in potentially related isolates, we see different patterns of assembly using the synthetic long read that we would from our expected SC5, 3, and 4 chromosomes. We can dig into this in a little bit more detail by pulling out this isolate here from student 880. And we can see that there's regions where there are potentially inversions and fusions between different chromosomes from the expected chromosomes and what we're actually identifying through the TEL-seq. So using that, we can identify regions where it looks like there is a fusion event between these 3 chromosomes, between 3, 6, and 4. We can design primers that would span that gap. And when we do genomic DNA extraction and just PCR to say, can we identify this band? We can identify a band only in that 880 isolate and not in that SC5, 3, and 4 reference. So this rearrangement has been validated by PCR. When we looked and went from that whole genome sequencing down to now we're going to look at some of the phenotypic variation, We see only minor differences in their stress responses. There is variability in whether the strains can grow or not grow, but it's not like a clear pattern where all of the oral isolates behave one way and all the fecal isolates behave another way, even under these different stress conditions. But what if we add the host? So not just these in vitro stresses, but in the presence of macrophages or a more complex immune system. So first, in macrophages, we could see that the morphology varies widely between strains. So these strains all were able to filament in the inducing Q of 10% serum and 37 degrees, but when we add that host, what we could see is that while the SC5, and 4 strain can have these long filaments inside macrophages, there's other strains that are kind of short and stumpy and staying as yeast after phagocytosis. Another point that I want to bring up that is, I think, important to say is that some of our invasion assays are used as a proxy for filamentation. But when we compare the filamentation capacity in the macrophages to that invasive agar, what we could see is that these isolates are able to make these long hyphae inside of macrophages, but in the invasion assay, only one of these is able to invade. So it tells us that potentially this is not a great proxy for what's happening in vivo, and speaks to the requirement for assaying your phenotype of interest in the system that you actually care about. Because of the relationship between morphogenesis and cell death has been really nicely elaborated on for SC5, 3, and 4, we then looked at all of our different commensal isolates for their ability to induce cell death in macrophages. And so these are bone marrow-derived macrophages that have been just added with Candida. There's no priming agent, no LPS. And what we see is that SC5, 3, and 4 is kind of the only strain that induces a significant amount of cell death. All of our commensal isolates look very similar to our mock infection, and even a set of clinical isolates that we're getting from bloodstream infections, these ones here with the P numbers, these also don't induce a significant amount of cell death. So it's potentially something interesting and weird about SC5, 3, and 4, and some of the things that we think about for being inflammatory might be only attributable to this one particular strain. Then we wanted to go from just a simple cell model to understanding virulence, and we're using the Galeria melanella waxworm model. So what I'm going to show you is the kinetics of mortality over time, the probability of survival. When we infect with our SC5, 3, and 4 reference strain, we see good mortality over the last 2 weeks. And then PBS, there's not very much. When we look at all of our human commensal isolates, what we see is that they're maintaining a high pathogenic potential. They're still basically all able to cause disease in this worm model. We have a couple that have a little bit less virulence, but they're still causing a good amount of worm death here. So this tells us that maybe these strains aren't making that same trade-off between invasive infection and colonization. But sometimes these monotypic infections aren't great at showing the differences between strains, and so it's easy. So a better way to look at some of these intra-strain fitness effects are to put them into a competitive model. So here we're doing a competitive fitness, again in the Galeria melanella model, and we're competing a fluorescently marked wild-type strain, which are going to be the dark spots here, they're sort of glowing, compared to a non-fluorescently marked commensal isolate from these students. And what we see at the end, after we grind up the worm and plate them out for CFU, is this differential representation in terms of fluorescent colonies versus non-fluorescent colonies. And we're going to use that to calculate that competitive fitness. So first, of course, we have to compete the wild type against itself to make sure that the fluorescent tag doesn't impact the competitive index. And what we see is that in general, the fluorescent tag doesn't impact, the competitive index is 0. There's not an advantage or disadvantage to having a fluorescent tag. And that means we can do this assay comparing to all the other commensal isolates. And what we got was actually quite surprising to us, is that consistently the commensal strains have a higher competitive fitness, even in the systemic model of infection. So they're still able to cause disease. They have high competitive fitness. Basically, these commensal strains, things that we're getting from healthy undergrads that are walking around with these strains all the time, they are still able to cause disease. So this wasn't quite the finding that we were expecting, but we still wanted to understand, can we use some of this genetic variation to understand phenotypic variation? Maybe we can't use virulence as our output, but we still have a lot of other phenotypes that we can use to understand the connection between genetic variation and phenotypic variation. So because we've done the sequencing, we can then look at all of the different variants between these strains. After we did extensive filtering, we still identified 100,000 high-quality variants across all 400 samples. 100,000 variants is too many to investigate. Is there a way that we can prioritize some variants that are likely to be interesting and give us sort of tractable phenotypes to track down? So here we took advantage of the fact that we had collected multiple isolates from the same individual. So we have student 814 gave us a lot of fungus. We had more than, I think we had 200 samples or 200 different colonies from this one student. But we ended up sequencing 7 of them. And we had both oral in black and fecal in pink. But what's important is that only one of these isolates had this hyperinvasive phenotype. So here, what I'm showing you in more detail is their colony morphology on YPD. And then after you wash it away, so you can see how many hyphae have invaded into the agar, in spider media, which is a more filamentation-inducing hue, and again, after you wash it away. And you can see that this particular isolate was the only one in that set that had a hyperinvasive phenotype. And so when we looked at the sequence of these 7 strains, we could identify only 12 coding SNPs that were different just in the invasive isolate compared to all of the other non-invasive isolates. And so rather than comparing this one isolate to all of the strains that are out there, by comparing it to a smaller set, we can sort of subtract away all of the variants that are not going to be informative. But 12 is still a lot of mutations to make. So how can we prioritize the next step and say which one to follow up on? So one of the things that we've been working on is the idea that co-expression is important and can help form hypotheses and decrease the number of tests you have to do to understand what a gene does. And so we've done this for Candida albicans, and we've also done this for Cryptococcus neoformans. And the idea is that if a gene is transcriptionally expressed with a cluster of other genes, that they always behave the same way across multiple different environmental and genetic perturbations, they're more likely to be involved in the same phenotype. Intuitively, that makes sense. Like if you, for example, are part of the ribosome, you want all of the ribosome genes to be turned on at the same time, so you have are maintaining stoichiometric ratios of that ribosome. So we built these networks for Candida albicans and crypto. This QR code here has a link to if you want to use these databases, but you can basically put in your gene of interest and it will give you your top co-express partners so you can make sort of maybe faster hypotheses about what a gene is doing. So we took those 12 genes with SNPs in them and did co-expression analysis, and one of them really stood out as a transcription factor. It was uncharacterized, it's called ZMS1, but in that set of co-expressed genes was a set of filamentation-associated signal transduction. So all of these genes are also associated with filamentation based on other people's studies. So we started to focus on ZMS1 and say, does this one mutation in this protein confer the invasive phenotype? So here, what I'm showing you are what we've picked as our invasive parental isolate, and then its nearest neighbor that didn't have that hyperfilamentation phenotype. When we do an allele swap, so we convert that ZMS allele from one strain to another, we could see that changing it to a threonine removes the hyperinvasive phenotype in that 168 background, so it no longer has that big halo. And when we make that into a serine in the non-invasive background, we can then convert this non-invasive strain into a hyperinvasive phenotype. It also appears to be a dominant negative because when we actually make the deletion of ZMS1, we don't see the same phenotypes as we do for these individual mutations that we're making. So this tells us that we can use the natural variation within a limited set to hone in on a specific mutation that's going to be causative for a particular phenotype of interest. So the summary for part 1 is that people are often colonized by multiple strains of Candida albicans. We don't just get one strain from our mothers and keep them forever. We can share them amongst your classmates. That there's extensive variation, including structural variation in the genome. There's a very plastic chromosome structure. And that if we have deep sampling of strains, not just like one representative isolate, we can identify SNPs associated with phenotypes of interest. And I think people who are working in the bacterial space are often using this for identifying sort of the population level resistance to antibiotics. And I think because we know that now people are colonized by these multiple strains, one isolate might not be predictive of the phenotype that you care about. And importantly, these colonizing strains can still cause systemic disease at rates similar to that clinical reference strain. So we're not really seeing that trade-off between commensalism and infection. And so that then brings us to the next question, like, why are we seeing this in mice, but not in humans? What are we missing in our models? So I want to go back to that original experimental evolution paper, where we took that reference strain S5E4 and 4 and colonized these mice over 10 weeks of experimental evolution. And then they get these consistently avirulent and yeast-locked strains. But what's important here is that this was done in the presence of antibiotics. And when they did the same experimental evolution in the absence of antibiotics, you don't get any of those phenotypes. And then recently there's a paper that came out that says, look, the competitive fitness of a strain really depends on that gut environment. So in the presence of antibiotics, the yeast-locked strain in pink consistently outcompetes the wild-type strain in black. But in the absence of antibiotics, that wild-type strain maintains its dominance, and that EFG1 is outcompeted. So this tells us that something is happening in that gut environment, depending on the presence of bacteria. So I'm going to bring Faith's picture back up, because this is a question that she has been focused on for the last few years of what are bacteria doing to Candida albicans? And what we're going to look at is the Candida albicans cell wall. So we know that that's the major point of contact between the host and the fungus. It has a very defined structure where there are these outer fibrils and manoproteins that are then locked into that inner cell wall, which is composed of beta-glucans and chitin above that plasma membrane. And because it's so important in regulating host pathogen interactions, it's been the focus of pretty intense study. And we know that there's a lot of different remodeling that happens in response to changes in the environment. So by changing carbon source, you can change the cell structure, the cell wall structure. By changing the oxygen, so normoxia versus hypoxia, you can see drastic changes in cell structure and inner and outer layers. Also in iron limited versus iron replete condition, again, you change the width of the outer cell wall versus the inner cell wall. And so Faith had a pretty straightforward question. Do bacteria influence this process? Is this enough of an environmental change to trigger that cell wall remodeling? And so what she found is that when she incubated Candida albicans with E. coli, you see this drastic increase in that mannan fibril. We can quantify each of these across multiple different TEM images and say when you add E. coli to Candida, it increases its mannan exposure. But not only does it increase the mannan exposure, it actually also increases the beta-glucan exposure as measured by dectin-1 staining, and the calcofluor white exposure as measured by, or chitin exposure as measured by calcofluor white. So these are the kind of flow plots that we're getting where we're looking at the population of cells and the intensity of staining, either for mannan, beta-glucan, or chitin. And then we take that mean fluorescence intensity, and then most of the rest of this talk is going to be using mannan as the proxy for this cell wall remodeling. So we see this change in response to, in these cases, all E. coli, but we also see it in response to other bacteria as well, both under aerobic conditions and anaerobic conditions, but to varying degrees. Because E. coli worked the best, we're going to use that going forward. So we know that when we change that cell wall, what we identified was that this actually changes immune recognition. So this is work in collaboration with Shannon Escher-Riggi and Kyla Ost. But if we incubate Candida with E. coli, it increases its ability to be recognized by dectin-1 and by dectin-2. So basically is at the level of the positive controls on both of these assays. And if we take our Candida that's been pre-incubated with E. coli and measure their ability to bind IgA from healthy human samples again, we could see that adding E. coli to our Candida decreases its recognition by IgA. So we think that this is going to be really important for regulating the dynamics of colonization inside the host. We also wanted to understand, how does Candida respond to bacteria? What pathways is it using? So we did a limited screen of transcription factors and kinase mutants to identify who doesn't respond or who over-responds to co-incubation with E. coli. And consistently, we saw that the HOG1 cascade is required. So deletion of HOG1, its upstream kinase PBS2, those mutants are unable to remodel to the same level as their parental strain. And that SCO1 transcription factor that's downstream of HOG1 is also unable to remodel. So we know that Candida is responding to E. coli. What is E. coli doing to induce that remodeling? So first, we took Candida and we're looking at, and then we can see if we co-incubate it with E. coli first, we have this drastic increase in mannan exposure. If we heat kill the E. coli, it doesn't do that remodeling. So that was important. It tells us it's not just some cell wall component of the bacteria that the fungus is sensing, because we know that things like peptidoglycan can change filamentation rates and other kinds of morphological changes. We're not really seeing that here. It has to be something live. And then if we take conditioned media, so if we grow E. coli in YPD, we'll say it's ECYPD, filter out that bacteria and then add it to the fungus, what we can see is that filtered conditioned media is able to induce remodeling. It's still able to induce the remodeling after that filtered supernatant conditioned media has been boiled, so we think it's probably not a peptide component. And we think it's not due to just that the E. coli has eaten all the sugar, because if we add back up to 2% glucose, that conditioned media is still able to respond. So it's not that the E. coli has eaten all the sugar and the fungus is responding to low nutrients. We also tried different carbon sources. And again, if we grow the Candida in dextrose, acetate, galactose, or glycerol, we don't see that induction of cell wall remodeling, so compared to this YPD control. And then if we add these different short-chain fatty acids that are sometimes known to be byproducts of bacterial metabolism, including acetate, butyrate, citrate, or lactate, we see some cell wall remodeling, but not to that level of the conditioned media alone. And because the HOG1 pathway is involved, and the HOG1 pathway is known to respond to osmolarity, we looked at the osmolality of the conditioned media. So we can see that we can induce cell wall remodeling with conditioned media from E. coli, Pseudomonas, Salmonella, Enterococcus faecalis to a lesser amount. And then also we saw a little bit of remodeling, but not a lot, in response to sodium chloride or sorbitol. So it's not significantly different. And we did a correlation plot of the osmolality of the solution versus the percent remodeling. And what you can see is that there's really no relationship between the osmolality of a solution and the ability of the strain, or the ability of the fungus to remodel. So this leads us to our foray into untargeted metabolomics. And so what I can tell you right now is that when we take conditioned media and fractionate it into either the aqueous or the organic fraction, only the organic fraction is sufficient to induce that cell wall remodeling. But we also know that all of these changes in the cell wall are probably going to impact other aspects of that interaction, not just the host. And we can really see this dramatically in terms of drug resistance. So here what I'm showing you are Candida albicans grown in either YPD or that conditioned media from E. coli. And we're looking at the ability of the strain to grow in an increasing concentration of drug. So we see that there is a slight change in response to amphotericin or fluconazole. But what was really dramatic is this drastic increase in resistance to caspofungin. And that really makes sense because we've totally changed that cell wall structure, and we end up with a huge amount of resistance to caspofungin, potentially impacting clinical outcomes. So our conclusion for part 2 is that co-culture with E. coli results in this global cell wall remodeling that impacts immune recognition, and that this requires a metabolically active E. coli and is mediated by a secreted factor in the organic phase. That Candida is responding to those secreted metabolites through the HOG1 signaling cascade, so it's a pretty central process for Candida. And that this bacterially induced fungal cell wall remodeling might also increase antifungal resistance. So we're hoping to investigate this further as well. So with that, I want to thank my lab. This work was really driven by Faith Anderson-Davis in my lab, and it would not have been possible without collaborators across U of M, the Bio 183 class, which generously donated their fungal strains. If anybody would like to have some of these fungal strains, please let me know. We'd be happy to share them and increase the diversity of strains that are being examined for Candida albicans biology. And with that, I'm happy to take any questions after the next talk.
Thank you, Teresa. I'll introduce our next speaker, and then we'll have the questions. It's my pleasure to introduce Dr. Chad Rappleye, who is a professor and chair of the graduate studies in the Department of Microbiology at Ohio State University. He started his research actually in a bacterial pathogen, Salmonella typhimurium, but during his graduate studies in UC San Diego, he got interested in eukaryotic cell polarization And during his postdoc in Washington University with William Gordon, he started exploring Histoplasma capsulatum as a Diamond Rainan Cancer Research Fellow. So his lab is focused on Histoplasma capsulatum and how this pathogen interacts with the host. And we are looking forward to your presentation on this topic.
Well, thank you, Ilian and Jay. Appreciate the invitation to share some of our work. Let me pull up my screen here and we'll get started. So as Ilian indicated, my lab is primarily focused on the pathogen Histoplasma capsulatum. Today, I'm going to talk a little bit more about what we're doing to understand the environment in which histoplasma lives when it's resident within a macrophage. Maybe as a way of introduction to histoplasma, a relatively less studied organism comparing to, say, Candida or Aspergillus. Histoplasma is a thermally dimorphic fungus, meaning that it adopts two different morphologies which are dependent on the temperature. So normally histoplasma exists in the environment as a soil-growing microbe. It's a filamentous mold that produces Candida, and as those Candida are released into the air and fall back into the environment, they just regenerate this mold form. In the laboratory, we can control this by temperature. So room temperature will induce the formation of the mold form. When, however, these Candida are inhaled into a mammalian lung, they experience a different temperature, of course, and that mammalian body temperature triggers those Candida to differentiate not into the filamentous form, but instead into the yeast form, which you can see in this image right here. The interesting thing about histoplasma is not necessarily just the difference in morphologies, but the morphologies themselves seem to have different fates in their lifestyle. So for example, the mold form, the filamentous form of histoplasma is completely avirulent if you were to put the mold into a mouse directly. On the other hand, the yeast form of histoplasma is virulent within a mammalian host. And so there's this distinction, not just of morphology, but also in the lifestyle that it holds. So once it's in the mammalian lung, histoplasma is taken up by resident macrophages. You also get a neutrophilic response as well as some dendritic cells. But the combination of these phagocytic cells engulf the histoplasma cells. And unfortunately, those are not sufficient to induce the death of histoplasma directly. So once you get a respiratory infection, there is a granulomatous type of immune response. Histoplasma generally is not well controlled, and you end up with extrapulmonary spread. So a couple things that I would just like to highlight here is that histoplasma is a primary pathogen, meaning that it will cause disease even in immunocompetent individuals. The second point is that during the acute stage of infection, histoplasma is nearly entirely within phagocytic cells. So just a couple of other comments on the clinical aspect of histoplasma. The degree of disease seems to be a function of both the dose of histoplasma that an individual receives, as well as the degree of cellular immunity that can be mounted by the host. So the majority of infections by histoplasma fall into this subclinical range, where healthy individuals with good cellular immunity, they inhale a dose of histoplasma, and during the innate immune response, the fungus proliferates within these phagocytic cells. But once you get activation of T cells and the subsequent production of interferon gamma and tumor necrosis factor alpha, the host switches to a situation where they can control and actually eliminate histoplasma from the lung and other sites of the body. So obviously, in an individual that has diminished cellular immunity, such as those with HIV, the fungal burden continues to escalate and end up with progressive disseminated histoplasmosis. Also, individuals that receive a high dose, okay, that is not controlled during that innate immune response can end up with a pretty severe infection that is clinically relevant under those high doses. But as I said, most of the time when histoplasma is infecting an individual, you do end up with disease, as shown here, kind of during this innate immune response. You'll have respiratory systems that are affected, as well as just general malaise. But healthy individuals will generally control histoplasma over time. So that takes care of the first part, that histoplasma is a primary pathogen. The second one is what I'm going to focus mostly on today, and that is histoplasma lives within the very immune cells that are designed to eliminate fungal pathogens. So this is just an image right here. These are macrophage cells Histoplasma is highlighted in blue at day 0. You can see that a number of the macrophages have been infected. But by day 3, those macrophages are just teeming with a number of histoplasma yeast because they've continued to replicate instead of being controlled. So the 2 questions my lab has been focused on are, first, how does histoplasma survive the encounter with host macrophages? And much of our early work established some of the virulence factors needed for that. But lately, our attention has returned to the second question, which is, once histoplasma has survived the encounter with a macrophage, how does it actually proliferate within these very immune cells? And this is an important aspect because many times we use different media in vitro to grow the organism and to approximate its conditions within the host. But as you look across the spectrum of fungal pathogens, things are going to be quite variable. So for example, pathogens like Aspergillus live primarily extracellularly. They're bathed in very rich interstitial fluids and so forth. Candida albicans has a variety of niches in the human host that it has to contend with, things that's different from the oral cavity, through the gastrointestinal tract, to even the surface of skin. And so these organisms tend to be very plastic in their metabolic processes. They adapt to lots of different environments. On the other hand, at this other spectrum, we have Histoplasma capsulatum, which lives within macrophages. And those same sorts of media don't really approximate what the environment is within the human host. And so I appreciate some of the things that Teresa was presenting today, that the environment really kind of affects what the pathogen looks like and how it functions. And so our studies are going to be looking at how does histoplasma manage the very different environment within the phagocyte. Specifically, right now, we're focused on questions about how does histoplasma acquire carbon, sulfur, and nitrogen when it's living within the phagosome of a phagocytic cell. This is an environment that is supposed to be generally restrictive for a lot of these environments. It's not a hugely rich media environment. So we just took a quick survey of what histoplasma can use to grow as carbon. This is showing down here. This is in vitro, just using single carbon sources such as different hexoses, as well as amino acids. And you can see right here, in contrast to some of the pathogens that can pretty much metabolize any carbon source you throw at it, histoplasma yeast are very restricted. So in our assay conditions right here, I'm just showing you a few of those. We can get it to grow on glucose. We can get it to grow a little bit on maltose, but galactose and sucrose and actually, to be honest, many other carbohydrates, histoplasma doesn't utilize very well. The other one that it utilizes well is, of course, amino acids, you can see here in the red curve. And so we started to ask the question, which metabolic processes histoplasma uses when it's growing inside of a phagocyte? And so we eliminated the functions of different metabolic pathways. Right here, you can see we've eliminated aspects of glycolysis, gluconeogenesis, as well as fatty acid utilization right here. And we looked at the ability of histoplasma to cause infections in a lung. We also did this in vitro. I'm just showing you the data right here, which manifests how histoplasma deals with it in the host. You can see that in wild type, after being inoculated, histoplasma can establish a very high fungal burden within the lung. If you eliminate the glycolysis pathway, there's no effect on it, suggesting that glycolysis is not necessary for growth within the phagocytic environment. Fatty acid utilization similarly doesn't seem to be required, but we do see decreases pretty significantly when we interrupt the process of gluconeogenesis. So this suggested to us, one, that hexoses aren't serving as a major carbon source during infection. Fatty acids are not being utilized during infection for growth and respiration, but instead it's another carbon source that feeds into the pathway, and then gluconeogenesis is necessary to generate those carbohydrates for life. So an obvious candidate for that, of course, is amino acids. A second indicator came from a mutant screen that we did for mutants that were unable to grow within the macrophage. So we identified a mutant in the gene called GDH2 that basically is involved in the catabolism of the glutamate, feeding into the citric acid cycle. So as I mentioned, this was from a screen, and this is just verifying it right here that histoplasma, after being infected into macrophages, grows nicely within those phagocytes. But in the absence of GDH2 and the inability to consume glutamate as carbon in the macrophage, it has attenuated the histoplasma growth pretty significantly. That translates into an in vivo infection. Again, this is a respiratory model of infection where we're quantifying the fungal burden wild type colonizing very nicely. But in the absence of GDH2 function, and in the absence basically of the ability to utilize glutamate, the yeast are severely attenuated. Now, interestingly, if we go the reverse direction, it's catalyzed by a second enzyme, GDH1. We eliminated the function of GDH1 through RNAi, and that showed no attenuation in vivo, suggesting that the metabolism of glutamate is actually catabolic in nature. So these indicators suggested that amino acids were serving a primary role for histoplasma growth within the phagocyte. And so we just surveyed the ability of histoplasma to utilize individual amino acids that might be found. This is a heat map right here showing you the ability of histoplasma to metabolize metabolize the amino acids as carbon, nitrogen, or sulfur. You can see here's glutamate right here. It serves as a very nice carbon source. It can also serve as a nitrogen source. But the take-home message here is that not all amino acids are efficiently utilized for carbon, nitrogen, and especially for sulfur. You can't even use methionine for sulfur. So this started us thinking a lot more about metabolism of the yeast cells, why they're resident within the macrophage. And through a very nice collaboration with members at Mount Alonso's lab, we started looking at the different metabolites of histoplasm when they're within the phagocyte. This is a very detailed slide right here showing you the different levels of metabolites. I'll just highlight to you the key here. The first 3 bars are histoplasma grown in vitro under different conditions, such as glucose or amino acids. The major one you want to pay attention to, though, is the very last bar right here in the light blue, which is what are the metabolites present in histoplasma yeast once they are within a macrophage. And I'll highlight 3 specific areas where we saw some interesting things. So the first one right here is the aromatic amino acids, tyrosine, tryptophan, and phenylalanine. You can see while tyrosine and tryptophan are modestly leveled within the histoplasma yeast inside of the phagosome, phenylalanine is quite elevated, suggesting that it might be taken up directly from the phagocytic environment. Cysteine is another example right here. While you do see some small levels of cysteine within the histoplasma yeast grown in different in vitro situations, it's highly abundant in cells that are grown inside the macrophage. Our final example right here is the production of leucine. You can see the precursors of leucine, looking at the dark bars, are actually really low, specifically 2-isopropyl malate. But leucine itself seems to be quite high. This suggests that it's not leucine in histoplasma is not being derived from synthetic processes, but is instead being consumed from the phagocytic environment. So again, strong indicators that yeast are utilizing amino acids within the phagosome as their carbon and potentially nitrogen source there. So to get a handle on that, we wanted to do a functional genetics study. But when you look at metabolic pathways, they're extremely interconnected, and it's very difficult to actually impair the utilization of amino acids with just a few mutants. So instead, we turned our attention to how does histoplasma acquire the amino acids that it's potentially using for its nutritional effects. And for this, we turned our attention to the amino acid permeases that are found in fungi. So this is just a phylogenetic tree showing the different families of permeases that have been identified. It includes things like amino acid transporters of the ACT family, some transporters that are found in the vacuole, some methionine type transporters, as well as the general amino acid transporter family, the YATs. And you can see here, there's a number of homologs that are out there. I'll draw your attention to a few of those that are present for histoplasma that are bolded right here. It has a number of general amino acid transporters, a number of others. Then it has some unique transporters here that have not really been assigned a functional group. Now, to get an idea of which ones might be important during growth of the histoplasma yeast, we compared the transcriptome of yeast cells, which remember are the pathogenic cells, versus mycelia cells, which are avirulent. And that gave us some really nice information. You can see right here in red with the yeast expression, there's really 3 major transporters that are expressed by the pathogenic yeast. DIP5, which is one of these general amino acid permeases, GAI1, which is a unique transporter you find right here on this clade, and then GAP3 also was highly expressed in the pathogenic stage. Now, to get an idea of the specificity of these transporters and which amino acids they would transport, we turn to a heterologous expression system in Saccharomyces cerevisiae. There's a strain of Saccharomyces cerevisiae which has been depleted of all the standard amino acid permeases. This is strain 22 delta 10 alpha. And what we did is we expressed individual histoplasma amino acid transporters within this cell, and then asked how well Saccharomyces cerevisiae can now grow on different amino acids as their metabolic source. And the growth is plotted right here. Wild-type cerevisiae, which contains all its amino acid permeases, can grow very nicely. In this case, we're showing growth on alanine. The mutant strain, you can see right here in the open symbols, doesn't grow well at all, even at millimolar concentrations of alanine. And then in red and blue are the growth of the strain when you introduce the histoplasma amino acid transporters. So you can see here that DIP5 seems to provide a pretty good level of import of alanine, GAP3 a little bit, but less so. And to get a semi-quantitative measure of this, what we did is we took the area under the curve for each one of those as an estimator of the ability of that transporter to transport that particular amino acid, and the results are summarized right here. So you can see, again, things fell out very nicely, that there were basically 3 major amino acid transporters. They actually had pretty broad specificity. You can see Guy1 right here transports nearly all amino acids pretty effectively. DIP5 a little bit more restricted, and GAP3, despite its name, the general amino acid permease, could really only transport maybe about 1/3 of the amino acids. But it did highlight that there was some redundancy. And so in order to perform our functional test, we realized that we were going to need to eliminate the function of not just one amino acid transporter that would transport one amino acid, but instead we're going to need to reduce the function of 3 amino acid transporters to really cover the spread of amino acid potential. So what we did is we used a combination of CRISPR-mediated deletion as well as RNA interference. And so right here, I'm showing you some in vitro growth of the mutant that we created on amino acids as its nutrient source, wild type shown here in black. You can see that elimination of a single transporter like GAP3 by deletion really doesn't affect the ability of histoplasma yeast to utilize amino acids. But if you eliminate DIP5 and Guy1, as well as DIP5 and Guy1 and GAP3, the triple transporter mutant, you can severely decrease the ability of histoplasma to utilize those amino acids for its growth. But this set us up a situation where we now have a mutant that cannot import amino acids and therefore is deficient in its ability to utilize amino acids. So what does that have as a consequence for histoplasmas to grow within the phagocyte? Surprisingly, it had very little effect on it. So down here, we're just showing the replication of histoplasma within the macrophage over 48 hours. You can see the wild type grows nicely, just as well as the triple transporter deficient mutant. But that was in vitro, so we also did a test in vivo, and we saw the same thing. Elimination of the ability of histoplasma to import amino acids and utilize those for growth does not affect the ability of histoplasma to establish infections within the lung, nor within the spleen. So it was kind of a surprising finding, since everything was pointing to utilization of amino acids. But upon closer thinking about this, we really only tested the ability of amino acids to be imported, and specifically, it was free amino acids. And as we thought a little bit more about this, histoplasma is occupying the phagosome with it, which is a degradative organelle. There are additional sources of amino acids other than free amino acids, and it got us thinking a little bit more about peptides as the potential source of amino acids. So to get an idea of this, we turned to an in vitro system where we digested some proteins, in this case hemoglobin and gelatin, into smaller and smaller peptides to see if they would facilitate the growth of histoplasma yeast. So first to note here is that histoplasma, the black bars represent hemoglobin in an undigested form as well as gelatin in an undigested form. And histoplasma yeast cannot use intact proteins as a nutrient source. However, if you use proteases like cathepsin D, trypsin, and protease K, you can see that now histoplasma is able to utilize some of those peptides produced as its nutrient source. And importantly, the more you digest that protein into smaller and smaller peptides, you increase the degree of growth of histoplasma. So this gave us some really interesting information that it's probably small peptides or oligomers that are providing the amino acids for histoplasma to use. In support of this, histoplasma has 5 peptide and oligopeptide transporters that will serve a different function than the free amino acid transporters. OPT9 and 8 are in one group right here, which are very similar to a number of the oligopeptide transporters of Candida albicans and other fungi, as well as 3 in the PTR family that you can see down here. So this is going to be our focus for the next while for functional genetics information. But in the meantime, we thought, well, can we supply it with a model peptide that would represent the intraphagosomal environment? But with the number of peptides that are possible with 20 amino acids, that becomes prohibitive. And so we wanted to use the idea of taking an amino acid auxotroph of histoplasma and using that as a probe for which amino acids are actually available within the phagosome. Again, amino acids probably coming from peptides. And so we've surveyed the metabolic pathways of histoplasma yeast. We've identified key proteins that are unique to the synthesis of specific amino acids. Just to give you an example of this, we'll talk about glutamine down here. Glutamine is created by the enzyme GLIN1. And if you take a look at the ability of histoplasma mutants in GLIN1 to grow in vitro on a substrate that lacks glutamine, you can see that it doesn't grow. However, if you provide glutamine, which is the red filled circles right here, you can see that you restore the ability of this auxotroph to grow. Okay? You can then take this mutant and you can use it in an infection situation. And if the growth or the virulence here of histoplasma is normal, it means that there's sufficient glutamine to provide for that auxotrophy that the mutant has. So you can see down here, during a lung infection, the glutamine mutants right here cannot grow very well at all. In fact, it was really hard to recover anybody from this infection. That tells you that glutamine was not present to rescue that auxotrophy when histoplasma was resident within the phagosome. And so that marks glutamine as being relatively unavailable, or at least not available in sufficient quantities. Now we've gone through a number of these mutants right now, and shown here in green are amino acids that are present within the phagosome to rescue the auxotrophy. In red, you can see those that are unavailable in the process in the phagosome to rescue growth. And so it's not as simple as just saying all peptides will have all amino acids, and that's what's present. It really does look like there are select peptides that histoplasma is metabolizing. Maybe that's select peptides that it can import, but select ones are being used for its nutritional source when it's within this phagosomal environment. So I hope I'm able to kind of explain to you how we're thinking about histoplasma metabolism, specifically what is available within the phagosome, how can we use that to better model our in vitro media and do a little bit more reductionist type of approach. It seems very clear that amino acid metabolism is linked to the ability of histoplasma to proliferate within the phagosome of the phagocyte. Our identification of the free amino acid permeases GAI1, DIP5, and GAP3 show that we can block free amino acid uptake largely, but the lack of any effect on histoplasma virulence really does indicate that there's a different source of amino acids rather than the free amino acids we were originally thinking. And this actually makes sense, right? Because it is a degradative organelle. There's things to think about. What is the source of the proteases? These are likely coming from the lysosome as it fuses. What are the sources of the proteins coming from? This may involve the autophagosomes, where it packages up proteins or other larger macromolecules that need to be degraded and deliver those to the phagolysosome for degradation. And what that implies is that histoplasma is actually exploiting the degradative nature of this environment to provide it with nutritional sources that it can use to really grow within this environment that normally is pretty hostile to fungi. So I appreciate the opportunity to share that with you, just to acknowledge those that have done the work. I'd like to thank specifically Stephanie Ray, who did the bulk of this studies as part of her PhD proposal, as well as Chen Shen, who was a postdoc in my lab and now has his own lab at Rhodes College. He really did the bulk of the work. And a lot of this has not been possible without the collaboration with Ana Alonso's lab at the University of North Texas, specifically Adrian Heckart and Celine McNee, which are providing us with the metabolic information, the ability to quantify some of these metabolites within intracellular histoplasma yeast. So I appreciate your time. I'd like to thank my funding sources, and I'll stop there and turn the time back over to Ilian and Jay.
Thanks, Chad. All right. So I guess I'll start. Several questions, Teresa, was around the issue of the E. coli and cell wall modification. So does it require cell contact or conditioned media is acceptable? And can you use potentially an E. coli mutant library to kind of maybe understand what genes on the E. coli side are responsible for that phenotypic change?
Yeah, so we have done a set of E. coli isolates, so not just our like the standard lab strain, but a sort of set of like UTI isolates and other things. And we see consistently that the E. coli are able to do this process. We've thought about doing a high throughput screen on E. coli mutants for their ability to induce the cellular remodeling, but it has been technically challenging. So we're sort of going at it through metabolomics, and then if we identify a target, then go back to the E. coli genetics and make those knockouts. So we see consistently that it takes about 3 hours to start to see that difference between non-incubated and incubated conditions where you start to see that separation. I think that's most of those questions.
Well, and also the conditioned media, does that have the same effect?
Yeah,
Or
yeah.
you-
So if we do when we have the E. coli conditioned media, so we're just growing E. coli and YPD. So again, YPD has yeast extract in it. We've also done it with LB, but like LB also has trace amounts of yeast bits. So we don't have like a pure E.
Yeah.
coli. I've never seen a fungal component to it, but we're thinking about an experiment where we try conditioned media from E. coli alone, or so like just E. coli or E. coli grown with Candida to see if there's a different amount of remodeling that occurs.
And is it unique to albicans, or do you see it in other Candida species, or also relevant to CF? Maybe the interaction of Aspergillus and Pseudomonas.
Yeah, so we haven't tried with we've tried it in the other Candida species, and in general, some of them do remodeling. Mostly the ones that are closer to albicans phylogenetically do more remodeling than the ones that are further. I think Oris was one that like didn't do it, but we don't have we haven't tried any of the molds or the sort of beyond Candida species.
Great. Thank you, Teresa. Get in.
All right, so the next question is to Chad. So there is some analogy with Candida albicans here. So it can grow on proteins, on amino acids, if you let it for 48 hours. So the question is, did you test, when you tested actually the gelatin and hemoglobin, did you actually use the overnight growth with normal media so that you activate actually those proteases?
That's a good point. We used just a yeast that had been pre-grown on kind of a standard rich media. It's glucose and amino acid containing. We did not specifically grow them on peptides first and then transition them to that. So it could be that maybe there is a protease induced. It's interesting with histoplasma, many of the transcriptional outputs are dictated by this switch between the mycelial stage and the yeast stage. So there are a number of proteins that are just induced as a process of converting to yeast rather than it responding to the host environment. But your point is well taken. Maybe there's a protease that is induced if you provide it with protein for a long time. It's a good point.
Thanks.
Teresa, so again, a few questions around the cell remodeling phenotype. So how long does it persist? And if you remove the signal, do the fungi revert back to the...
Yeah, so we haven't actually tried that experiment yet, but we will. So we don't know.
Cool.
Yeah,
And
we only know the induction, not the reversion.
So I was a little bit surprised to see the same activity with Pseudomonas, given that Pseudomonas is so genetically divergent from
Yeah,
Enterobacterium.
it's definitely not as strong with Pseudomonas as it is with E. coli. Part of it could be because our E. coli are growing better, and so potentially producing more of the metabolite. We definitely, there are differences, and it's also possible that, there are multiple pathways that are involved in this process, and they all look like remodeling to our eyes, but they're through different pathways. So I won't rule out that there is specificity, but I will say that there are some differences between the species as well.
And then what do you think would happen in an anaerobic environment?
So we still see the remodeling in response to some anaerobic bacteria. We actually started this project looking at Bacteroides theta iota omicron in that anaerobic condition. So the baseline remodeling levels are different, but we still see that they induced the remodeling, but like in comparison, B theta is like much worse than some of the aerobic conditions. But if we take E. coli, even in anaerobic conditions, E. coli grows better than B theta, and then we just have a different level of response. So I think it's something pretty general.
Thank you. Julian.
Okay, so Chad, have you used some inhibitors to block basically the activity of acid-activated lysosomal proteases? So here the suggestion is lysosome inhibitors or VATPase inhibitors. And is there some connection also with autophagy inhibitors? Have you looked at that?
Yeah, coming at it from the host side, right? You're thinking exactly as we're starting to think about this as well. Those are perfect targets. One thing that we have investigated preliminary is if we could eliminate the function of host proteases. So, for example, can you treat them with inhibitors that block, say, the cathepsins or other metalloproteases and serine proteases? And so we've tried to do that with just some broad spectrum protease inhibitors. We had one result that was suggestive that if you decrease protease activity within the host cells, that you actually reduce the replication of histoplasma within host cells. That would be consistent with this model that the host is providing the necessary peptides through breakdown of proteins. Upon repeat, that didn't show so well, and we're not sure if that's really going to give us an answer because those proteases can be, or the protease inhibitors can affect a lot of broader processes. And so, like I say, I really like where you're thinking because that's where we're going as well. If we can identify more of a specific aspect, I like the idea of phagolysosomal fusion, and there are a number of autophagy inhibitors now that are relatively specific. So I think those are really great areas to venture forward in.
You're even going maybe with some autophagy and knockouts,
Mutants.
the regular mutants that could so
exactly.
you come around hitting both for
Yep.
some
And the availability of mouse models I think is going to be really nice in that aspect.
Yeah. Right, thanks.
Great. Teresa, so there was a technical question about I think you picked 6 isolates to do a deeper dive on. And how did you pick those 6? There was a question.
We picked them because they looked weird. Where we had enough isolates from one person that we had already sequenced where we could say like, well, here's one weird one and here's a good set of comparator ones. Basically, we realized that we wouldn't be able to do any kind of SNP analysis for all 46 because there's too many variants.
Yeah.
And I think, if we were able to sequence all 900 isolates, we'd have a better sampling set to pick isolates from. But we picked them because we had already picked isolates to sequence based on sort of trying to get matched sets of here's some weird ones, here's some regular ones. We were enriched in our ability to do that kind of analysis. But I'm hoping that as sequencing costs decrease and we sort of have a bigger breadth of samples that are analyzed, we'll be able to do that kind of analysis in a more unbiased way. So this is something we're thinking about for Candida auris, where there's like hundreds of genomes that have been sequenced versus albicans. There's actually quite a limited set because people have mostly been working with that reference strain, which has been super useful because we can make knockouts and do comparative analysis. But I think the genetic background matters, and we're not really leveraging that natural diversity to dig into some of those phenotypes. So we picked them because they were weird, and that was great.
And then a related question regarding the first part of the talk in terms of the chromosomal recombination of or inverse that you noted in the commensal isolates. And the question revolves around, do you think this is potentially an evolutionary advantage, and this is potentially a fundamental way that potentially the Candida adapts to different environments?
So I don't know if they're doing rearrangements in response, in response to a particular stressor, right? So if we think it's adaptation in response to something, then we would expect that after induction in a queue, you would see it. I think there are fragile sites in the genome, places where there are these repeats. I think some nice work from the Selmecki lab has said like these long, these repeated regions allow for amplification of a particular region because of the ways that it resolves these like holiday junctions.
Mhm.
So it's possible that's occurring in albicans too. Potentially that's going to change the dynamics of like which chromosomes become aneuploid under the stresses because it is sort of recombining away different genes from each other, right? So consistently, you would normally see that chromosome 5, I can't remember which arm it is, but amplifies in SC5, 3, and 4 when you do that fluconazole stress. But if those genes are no longer all on the same chromosome, do you still see that same amplification? So I think there's a lot of potential there, and it's going to change the evolutionary trajectory based on genetic background. But I think that's a fundamental principle of evolutionary change.
Thank you. Good.
So the next question to chat. So does this mechanism apply to other intracellular pathogens and other fungal pathogens? And here the premise is maybe different peptidases will be taking place and will be utilized in that situation. And I can add to that, if you do co-competition basically with intraphagosomal fungi, what will happen?
Interesting idea. I think the challenge here is you would need to have fungi that are more matched in their proliferation rate. So it takes, histoplasma on the order of 8 to 10 hours to divide. And so it's a relatively slow growing fungus. It's really unclear why it's so slow, and it seems like every other yeast out there grows very, very quickly. And so, for example, if you were just to take Candida and do it 1-to-1 with histoplasma, let them get phagocytosed, what would happen? I think the lifespan of Candida within the phagosome is much lower. It's much smaller before Candida will actually lyse the cell or get controlled itself. Whereas histoplasma is kind of setting up for this really long term, I'm going to stick this out, I'm going to basically make this my niche, and so I need to exploit it as best as possible. So I think the challenge would be you'd have to really try to match those sort of things. On the other hand, I think that the work, and it makes sense, right, that there are probably a number of peptides that are available pretty quickly when a phagosome is formed. And whether other fungi can exploit that or not, I think that's one of the questions that's actually out there and remaining. I know amino acids have pretty profound effects on Candida yeast. Aspergillus, again, it's a little bit longer term in there, but it's not, there's a long process of germination. So that may have some different effects. So I hesitate to say there's a lot of comparison between fungi here because the intracellular times of these fungi vary widely, as well as what aspect of their growth they're actually going through. Cryptococcus might be another one, right? It definitely gets within the phagosome or the phagolysosome of phagocytes, stays there for a good little bit of time, but then it undergoes this process of vomocytosis. And so that one may serve more of a common mechanism because it hangs out within the phagosome on the order of hours instead of, maybe just, tens of minutes or short hours lifespans. So I think what we're going to find is that different fungi exploit it for different reasons, depending on what their ultimate lifestyle is supposed to be.
Thank you.
Teresa, we had two questions around the issue of trying to assay virulence either in murine models or waxworm models in correlation with the humans and particularly in isolates that may be derived from non-mucosal surfaces like blood isolates. Are there any? How good are these models at predicting human virulence and
Yeah,
are there good assays there?
I think they're definitely imperfect. We were not able to do all of the virulence assays in mice based on like what the capacity of my lab, but I think all models are flawed in some ways, and then the question is, when do they become useful, and when do they tell you something that you might be able to explore in a more systematic way? It's still unethical to put Candida into people at the levels that they would. Even in our mouse model, we put a large number of Candida into the IV, and whether that mimics natural infection is also unclear. The type of mouse and like the immune status of the mouse, does it mimic people like all of these models are going to be flawed in some ways, and we're just trying to figure out. I think that's why we really liked using those human samples to say, like, are we seeing that same pattern from human samples that are collected without damage to the individual compared to these mouse models? And so potentially we're seeing something that's totally artificial related to the worm, but it's not what people have expected to see based on their previous results. So I think there's something still useful in that information. But yeah, the worm has limitations for sure.
Thanks, Gillian.
So the next question, the phagosome is the degradative organelle, but in the past models, phagosomal fusion was not thought to occur depending on histoplasma method of entry. Would some of the small peptides be expected to be taken up with the yeast? And do you have any insights on the carbon source during sporulation within the microphage and then the transition from pathogenic to non-pathogenic state?
Okay, so the first question, whether I think centers around whether phagolysosomal fusion occurs in different cells. Yeah, and so it depends on the species, and a lot of this is in vitro modeling that certain cells will house histoplasma in a phagosome. Some will house them in more of a phagolysosome with lysosomal fusion, of course, occurring. The macrophages that we used for this initial study are some murine cells that we confirmed that phagolysosomal fusion does occur in. And so that kind of may gear your thoughts around that. I think it's actually a really interesting question that this may actually be an avenue for whether a particular phagocytic cell is susceptible to histoplasma infection. You know, if it has a very low rate of phagolysosomal fusion, maybe there's lack of delivery of sufficient peptides, and that may be a means of better control of this intracellular pathogen versus cells that just take everything up, use it with the lysosomes, and in so doing, provide this primary pathogen with the nutrients that it needs. So I think it's a great question. I think once we get a better understanding of what peptides might be utilized, we can ask some really more specific questions about, is this happening in a murine cell at the same degree as a human cell? Is it happening in a dendritic cell to the same degree that it's happening in a macrophage? So I think it's really fantastic questions. I think we just need to establish a little bit more information at the baseline before we can get those directly. And your second question was something about sporulation.
So an insight about the carbon source during sporulation within the microphage and then the transition from pathogenic to non-pathogenic state.
Oh, okay. Yeah, okay. So sporulation, so in a organism that is alive, right? So the temperature is maintained, the nutrients are there. Generally, histoplasma is going to undergo this replication process where it just continually grows and buds off yeast and grows as yeast, so forth and so on. You really don't get sporulation occurring until the temperature goes back to that more environmental condition which triggers it. So as long as the mammalian host is maintained 37 degrees, you're really not going to get any histoplasma yeast converting back into mycelial phase during an active infection. However, the model is, right, once the host dies, histoplasmas within these cells, they're going to drop in temperature, they're going to drop in function, and then histoplasma goes back into its filamentous form, which can then spread and well, it undergoes the conidation effect and then spreads. I think it's really feasible to think the same sort of things might facilitate this conversion. People haven't studied sporulation extensively, particularly from a metabolic aspect. We have a number of media that we use to induce sporulation in vitro. Have those been rigorously validated that those are the actual cues? At this point, I think a lot of times we're just saying it works and we use that for it. So I think with all these questions, the main thing is trying to mimic the actual environment in vitro as close as possible. And that's the challenge, right? So many times we use rich media because we get great growth of the organism. It's usually glucose-based. And then as you look at it closer, those are probably not the same signals or the environmental conditions that the organism is experiencing during infection, whichever it might be. For histoplasma yeast, a glucose-rich media doesn't seem to be appropriate at all. For sporulation, we really have no information about what are the true inducing signals for histoplasma sporulation. But it raises that same question. Once we understand more, we can do more of our in vitro media to reflect that.
Interesting. A lot more to do.
Mm-hmm.
Teresa, do you also see remodeling in hyphal cells, or is it more predominant in the yeast one?
Yeah, so we've tried to do this where we induce the hyphae first and then do the remodeling. It ends up that we're still working on getting the technical aspects of that set. Like we're losing some hyphae because they don't centrifuge as well to do the microscopy with. Most of our assays are done with flow, and that they don't really flow very well. But I think that's an interesting point of like, if they are already hyphal, does that change the remodeling dynamics? Definitely a lot of the transcriptional signatures that we're seeing are associated with cell wall remodeling, not necessarily with hyphal formation. So we don't see the induction of hyphae the same, like we don't see a huge induction of hyphae. We see mostly this remodeling, and they're all still as yeast. But yeah, I don't think we have a good answer yet for if they're already hyphae, what happens to that next step.
And then Kenya Honda had a paper in Science a few years ago showing that patients with IBD are often colonized with these oral unencapsulated E. coli strains. So maybe have you had it? Are your E. coli strains encapsulated, or have you looked at the to see if Candida is remodeled in patients with IBD?
Yeah. So one of the things that we don't have is a comparative set of like, are the strains that cause IBD different from the ones that are in healthy individuals? And I think that would be an interesting set to look at too. I know Ilian's lab has a bunch of these strains as well. We haven't. I think all of our E. coli are encapsulated. We don't have any E. coli mutants that we've tested, but it's possible that that's important. It's a good question. We can see if we can find one. I'm sure there's people in the department that have some of these strains.
All right, Ilian.
So there is some analogy here with Mycobacterium tuberculosis because it behaves similarly to H. capsulatum. So have you thought about this direction? Is MTB although MTB actually inhibits acidification in the phagosome, does H. capsulatum manipulates this process, for example?
Yeah. A lot of times we like to throw the comparison that histoplasma is essentially the fungal version of tuberculosis.
Yeah.
There's a lot of similarities clinically, granulomatous disease, long-term resident phagocytes. There's even aspects of histoplasma biology that it can stay latent, like TB. But specifically for the phagocyte, histoplasma does modulate the pH of the phagosomal environment. It's unclear exactly what mechanism it's using to do that. There's been some reports that it simply involves the decrease in the number of vacuole ATPases that pump protons into the lumen there. On the other hand, there's some aspects of histoplasma biology itself that may be contributing to that. For histoplasma containing phagosomes, the pH generally lies somewhere on the range of about 5.5 to 6.5. So it's definitely not as acidic as a fully, matured phagosome. It's definitely not as neutral, and it's thought that facilitates the ability of histoplasma to acquire iron. So there's this common feature. Is it doing it functionally the same way as Mycobacterium tuberculosis? I'm not sure. There's also reports of tuberculosis using fatty acids as a growth substrate, particularly in acquiring some of those lipids and so forth that you find within the alveolar fluids. And from our work right now with the mutational studies, it doesn't look like histoplasma can even utilize the fatty acids that might be found. So common environments, they might be exploiting different nutrient sources.
Yeah.
All right, Teresa, so one question about the clinical isolates from the students. So, Michigan's a state school, but presumably not all the students are from Michigan. So did you see any clustering from, the geographic origin of the student?
So I don't have that information. That would be theoretically identifiable information of those students.
Yeah.
I assume it's a pretty wide range, and based on the people had seen some association between certain strains of certain clades of albicans in certain geographic regions, and based on that pretty wide distribution of our isolates from different clades, probably a wide range of student origins as well. But the fact that the students are able to share strains with each other or have multiple clades from different clades of albicans that are theoretically from different geographic regions suggests that there might be more movement than expected.
Yeah.
But
And-
yeah.
And then have you assessed the IgA coding in from the oral versus the fecal isolates? Do they have equivalent amounts of IgA on them? And
you know, I don't think we've looked at that systematically. Kyla Ost is working on a lot of that biology. So she has all the strains, and we'll see whether we can see a consistent pattern. Potentially,
and also maybe whether IgA coding affects the interaction with the E. coli, right? So,
yeah, so we haven't like pre-coded and
Right.
then E. coli.
All right, I'll turn it back over to you.
Okay, so I think I'll go with the last question here because the time is up. So actually, I hijacked one of the questions with adding my question at the end. So let's go back to that. So the question was so they were referring more to the metabolism of Candida and macrophages to yeast with the phagosome. So it wasn't actually on the host side that much. And if that initial nutrient requirements are the same as observed with established pathogenic yeast.
So the question is, does the environment of the phagosome nutritionally affect the ability of Candida to convert into yeast? Is that?
Yeah, so the metabolism of Candida in the macrophages twists within the phagosome. So
Yeah,
the
okay.
comparison
So
there, that's what they meant as well.
Yeah, so
And I went to competition and stuff like that. So,
yeah, the metabolism of Candida into yeast is not known at metabolite levels. Other than a few, you do see some elevation of things like cyclic AMP, which it looks like it's serving more of a signal transduction type of effect. I mean, clearly germination of Candida is going to depend on what the environmental factors are. Phagocytes can take up Candida just as well as they can take up yeast cells. And so, you know, they may occupy the same, we'll say, nutritional environment because they're both going into a phagosome. On the other hand, the mechanisms by which they're taken up might differ. And will that lead to differences in the phagosome? I think this is, you know, a huge question that's still out there. These sort of studies are just beginning to emerge. Can we actually profile the phagosomal environment? We've done pretty good about saying what are the proteins that characterize these different compartments, but some recent work from David Underhill was starting to get at, you know, whether these pathogens recruit the same sort of factors to the phagosome. He was looking primarily at bacteria versus fungi, but even within the fungal world, depending on the mode of entry, I think that the phagosome might vary a little bit. So I think it's a great question, and definitely I think the nutrient composition of the phagosome can influence the germination of the Candida. Definitely.
Thank you.
All right. Well, I think Dr. Ilyev and I would like to thank both speakers for excellent talks today, and I think we kicked off 2025 with a great session of MycoTalks.
Yes, thanks everyone. It was a fantastic session, and I hope everyone enjoyed.
Yeah. See you in February.
Yes.
Okay.
Bye now.
Thanks, everyone.